Welcome back to Berkeley Optometry's live online CE series on COVID-19. Our first lecture this afternoon is an update on COVID-19 in the eye presented by Dr. Suzanne Fleiszig. As a reminder, recording or capturing of images in this session is strictly prohibited. Handouts will be available after the completion of the lecture. All participants' names, emails, and login times are automatically registered when entering Zoom. All participants are automatically muted upon entry. Questions for the speaker should be entered into the chat box, as well as if you need tech help. I'm pleased to introduce Dr. Suzanne Fleiszig. Dr. Fleiszig received her OD and PhD degrees from the University of Melbourne in Australia. A professor of optometry and vision science at Berkeley Optometry, she also holds appointments in the infectious disease and immunity and the microbiology programs at UC Berkeley, and is an affiliate of the Proctor Foundation at UCSF. Her research program focuses on the pathogenesis of contact lens-related infection. Dr. Fleiszig has served as the president of the International Society for Contact Lens Research and the vice president of the Tear Film and Ocular Surface Society. Currently, she is the chair of the Council on Microbial Sciences for the American Society for Microbiology, and in that role recently organized an international summit focused on urgent priorities for COVID-19 research. While she has spent the past 30 years in the USA, she has not lost her Aussie accent. Here is Dr. Fleiszig presenting an update on COVID-19 and the eye.
Hi, everybody. And I apologize for my accent. Those of you who actually were in my class will know that I teach in the first year of our optometry program here, Anatomy and Physiology. And our students usually takes them about three or four lectures before they start tuning in and understanding anything I say. But hopefully those of you who have already been through that won't have too much trouble today. Okay, so before I start, I should just talk about disclosures. So I don't have any financial disclosures related to this lecture, but, and actually we have started to do some research into COVID-19, but it's not funded by any companies, in fact, not even funded by the NIH at this point. I'll be talking about that later in the lecture. My usual research program is focused on bacterial infection, and that is funded by the NIH, although in recent years we've also had some unrestricted gift funding from Cooper Vision. So I thought I would let you know that, so you can consider it in terms of some of the things that I'm going to talk about today. Okay, so I put this slide in, and when I did that, I didn't realise that Dr. Harvey was going to do such a fantastic job talking about the epidemiology and about the differences between this and the flu. So I'm going to really skim over this particular slide. Just a couple of points. First of all, that this virus is a lot more contagious and is deadlier than the usual flu. So Dr. Harvey talked about the 1918 flu, which was a very different situation. But when we talk about the regular flu, this is actually a worse problem. The other thing that I should probably mention, going to the bottom of the slide, is that whilst this thing is not as deadly as SARS or MERS, which were the other two coronaviruses that we've had to deal with in recent years, and not as deadly as the 1918 flu, this is probably not going to be the last one of these coronaviruses. So the experts in the area are telling us that SARS-CoV-3 is coming soon, and there's going to be others, and they're going to come more and more often. And the death rate for cases of SARS-CoV-2 is actually, as Dr. Harvey mentioned, somewhere between 1% and actually some experts are saying up to 5%. We're not actually going to know that number until this is all over. And that's because we don't really know exactly how many cases there are, and also because some of the cases that are out there today have not come to fruition. So in other words, the death rate from the people who are currently infected, we don't know yet. So the numbers are really up in the air. But we do know that the death rate's not as much as SARS or MERS. And so what we don't know is the next coronavirus that comes along and behaves like this one could actually be even more deadly. So we got lucky with this one. So I think some people are actually seeing this as a warning that we need to get more prepared for the future. So I just thought I'd just bring that up because that's a very important point. Okay, again, some of this was covered by Patsy Harvey this morning, so I'm not going to spend too much time on it. I put these questions in the last time I gave this lecture. And, you know, we had no answers to these at that point, but I think some of them are starting to become clearer. So first one, is this seasonal? Well, it looks like it's not. As we can see, it's still going on. The infection rate now is higher than it was earlier this year. Perhaps it's going to get worse in the fall. That's still to be seen, but it's certainly not going away during the summer. Will we develop lasting immunity? Well, okay, this morning I would have told you that there's a concern there. And that is because a study came out last week that showed that the antibodies that are being produced by people who have had this infection are actually not lasting very long. So in a lot of people, antibodies disappear after about 2 to 3 weeks. So that was looking like a real concern in terms of us developing lasting immunity. What I just saw this morning, and this is going to tell you just how fast things are moving in this field, is that you prepare a lecture and the next day it's all changed. A study just came out and it's showing that people are developing cellular immunity. And that's actually really good news because cellular immunity tends to be much more long lasting. So this is very preliminary. And in fact, this paper's not even been peer reviewed. It's just been posted. It needs to go through a peer review process, but it is good news suggesting that people are starting to develop cellular immunity, which will be more long lasting. Okay, so good news today. Okay, this is a concern for me and many of the experts in the field is, but what happens if this thing circulates through billions of people? So we have all these people in the world who have got no immunity to this to start with. And so this is why it's spreading around so quickly. So viruses are able to mutate. And even though the relative mutation rate of this virus is very low, the more people it circulates through, the higher the probability that a mutation will arise that could make it more virulent. It could become less virulent, more virulent, but actually what happens is you don't just get one mutation going in one direction. Often there'll be many different mutants spreading around different places. And these are called clades. So you have like clusters of virus with different mutations. Well, this is the question we're all asking. And testing and social distancing are only delaying things. None of us started out with immunity to this virus. It's brand new, unlike the flu, which has been circulating for centuries. We have some underlying immunity to the flu. We're all completely naive to this virus. And so we can hide away, but as soon as we come out, it could infect us. And if it actually gets around the entire US population, even if you consider the death rate only to be 1%, and some people think it's higher than that, then that predicts that up to 2 million people could eventually die just in the USA. So, you know, the hope is that's not going to happen because of, you know, mitigation, you know, maybe therapies, better management. You know, we might be able to reduce the death rate by not just therapies or vaccines, but even the way we handle people when they're sick. So, you know, the longer we can postpone the ultimate conclusion of this, the better probability there is that that isn't going to happen. Okay, so so many things that Dr. Harvey talked about this morning where I had actually read different things, and even experts conflicted in what they believe in or what they're predicting. And, you know, why is this confusion out there? You know, why is it that we can't just answer all the questions that we're asking? And that's partly because the epidemiology is still really unclear. And I think you kind of got that message this morning from Dr. Harvey's lecture. You know, there are these asymptomatic carriers, you know, we've got the variable incubation time. You know, it looks like some people are getting recurrences of this. You know, we're not testing enough. And then, as I already mentioned, you know, in many people, the antibody response is short-lived. It only lasts for about 2 or 3 months, or sometimes weeks. So that means if you test someone to see whether they've had the infection and they don't show an antibody response, you think, oh, well, they've not had it, but in fact, they might have had it, and you just don't know because the antibody response is gone already. This is also a huge issue, and that is that there are giant gaps in our knowledge about not just the virus itself and how it behaves, but also how our immune response deals with this virus in a positive way. You know, the people who actually recover well, who are asymptomatic and actually completely fight it off successfully, we don't really understand how that works. And there's also the inflammatory response that is actually the thing that eventually causes the terrible lung disease and kills some people. So we don't know enough about that yet either. So there's 3 giant topics here where the information just isn't there that we need to be able to really get a handle on this. And that's, you know, obviously because it's a brand new virus, and there's just not been time to do this. Okay, so we're all hanging our hope on a vaccine. And there's been a lot of talk about, you know, when is the vaccine going to happen? And some people think it's going to be soon. Well, the answer to that question is that actually we may not necessarily be able to get a vaccine. And in fact, we don't have vaccines for other things that have been around for decades or centuries, despite decades of research. So, you know, we don't have a vaccine for HIV. We don't even have a vaccine for the common cold. In fact, some of the common colds are actually coronaviruses. And I can tell you there's been a lot of effort to try to develop vaccines against those, and nobody's been successful. Same thing with the flu. Even though we have a flu vaccine, it's only an annual thing. We don't have a universal flu vaccine that you get once and protects you for your life, the way we have for some other viruses. And so, you know, that means that, you know, we could be doing a lot of research and trying to do it quickly and whatever, and we just may not be able to actually find a way forward. And this really does depend on the type of immunity needed. And that, as I said, we still don't understand what the immunity is for this. But even if there is a vaccine, you know, another concern is that this vaccine might not actually help the most vulnerable people, because people who are older are the most vulnerable, the immunocompromised. These are people where, you know, vaccines may not be as effective because they don't mount a strong immune response. You know, this is often, you know, why vaccines are given to children, younger people. And also we have to tread very carefully because, you know, you can have negative side effects. So, you know, there are safety issues. A lot of testing needs to go on to make sure you're not causing more harm than good. So that process needs to unfold. And for each vaccine, there's a protocol. You know, you know, some vaccines, you need boosters, you need the right dose, it needs to be administered the right way. And if you don't do it the right way, you can kill someone. So, you know, even with the existing vaccines that we have that are very successful now, if you give it to someone at the wrong age or in the wrong condition, administer it the wrong way, it can be dangerous. So all of that takes time. Having said that, there are currently, the last time I looked, and that could be different now, 13 vaccines in clinical trials, and there's 128 total being developed. Fortunately, because of SARS and MERS, we got a head start. So there was some some vaccine development already going on for SARS and MERS. And because they're very similar coronaviruses, the people now working on SARS-CoV-2 have been able to leverage the knowledge and information and techniques from that earlier research. So the goal of this vaccine development is to have something that generates antibodies that last, and preferably also T memory cells. So that's the ideal. If you can actually get those memory cells, then you get a long-lasting vaccine. And now doing the second thing, the T memory cells, is much more challenging than a vaccine that's more just antibody generating. So there are various approaches that are being taken. One is the classic approach, which is to take the virus and attenuate it in some way so it's not as virulent, and use that. You know, either a live virus or a killed virus, or you can just use antigens from the virus. So just the parts of the virus that actually stimulate the immune response. But there are other approaches that are being used, and this is thanks to all the research that people have been doing for many years, studying immunology and developing vaccines for other things. So one approach is to use genetic engineering, where instead of using the virus or parts of the virus, you actually take the DNA, so the genetic code or the RNA for those viral antigens, and you use that, and then that produces the antigens in the body that stimulate the immune response. And so this can be done either, you know, with the real DNA or an RNA, or, you know, people are using recombinant methods. They're also attaching things to adenovirus. You know, and that's you know, an approach that's been used in research for many years. Another interesting thing is one approach is to use the measles vaccine, where they're taking antigens from COVID-2 and putting it in to the measles vaccine, so that at the same time as the person's being vaccinated with measles, they're also mounting an immune response to viral antigens against SARS-CoV-2 as well. So that's, I think, a really interesting approach. Another thing that's being studied is boosting general immunity. So looking at other vaccines that are already existing and known to be safe. The polio vaccine is being tested, and also the BCG vaccination, which is for tuberculosis. And in fact, in some countries, people are already vaccinated with that. And so, you know, it'll be interesting to see over time how things go in those countries. There's also research going on which is aimed to speed up the development and a process, you know, to speed up the research technologies so that in general vaccines can be developed faster. So that's going on in the background. And then there's also many efforts being put into increasing the speed at which things can be produced. And so all of that's also going on in the background along with the vaccine development. So, you know, every day there's something going on in this area, and it's really just hard to know how this is all going to pan out. Okay, the other thing that's going on is a lot of crazy amount of activity, people trying to develop therapies. And actually we are also doing this, so I'm going to talk about this a bit later. So how do you develop a therapy? Well, there's three different approaches you can take here. So one is to attack the virus. All right, so that's you develop an antiviral. The second approach is you can bolster the person's immune response, you know, so you help the immunity along to get rid of the virus. The third thing you can do is limit that damaging inflammatory response that happens in some people. So there's three different ways we can go, and there's research going on in all three of these. Probably a combination of all of them is going to be the most effective. I'm just going to talk about a couple of things that are going on out there that you might be interested in. So in terms of antivirals, you've all heard of remdesivir, which has been approved. The problem with that currently is that it has to be delivered IV. So that means the person with the infection has already got to be in hospital and treated with IV. So, and this is an antiviral. So it would be much better to use an antiviral earlier, before the person actually gets to the point that they have to go to hospital. Kill the virus before you get to that terrible inflammatory disease. And so, you know, that was a problem. But what I found out last week is that they're now developing an inhalation method, which I think is really exciting, because that's something somebody could use at home when they're sick, you know, an inhaler. And also the other thing is you're introducing that antiviral directly into the lung, and it's not going to go systemic and have other possible side effects. And you're going to be able to get a higher concentration right where it needs to be. So I think that's a really exciting thing. And you know, there's evidence in remdesivir, you know, is very effective. So this could be really cool. And this is not the only one of these promising antivirals. There's a number of them that are being tested. So one of them, Merck is currently testing. It's an oral antiviral. It is just called EID 2801 at this point. It still needs to be tested in humans, but it's been tested, I believe, in animal models. So another exciting thing. And then UCSF, I'm going to talk a little bit about their research program, which is just unbelievably exciting. They have something that's already in phase 2 trials. That's an antiviral. I can't say that, particularly with my accent. You know, it's, I don't know why they gave it that name, but there it is. And then another thing that is also, I think, pretty interesting is that there's some already FDA-approved drugs and also some drugs that are already in trials for other diseases, particularly some of the anti-cancer therapies. And so this is one that's looking promising in that arena. Again, I can't say that either, but you know, you can read that. So yeah, so there's a number of things happening in the antivirals, and I think that's going to be the best approach, is to use an antiviral, get this early before the person ends up with the serious lung disease. Okay, so bolstering immunity, also some really promising stuff going on there. Convalescent plasma. The research has shown that convalescent plasma, so this is taking plasma from someone who's already had the infection and has recovered if they have antibodies. And they've shown that you can actually reduce the 7-day mortality rate from 15% down to 5%. So very, very promising looking. The development of monoclonal antibodies. So this is instead of using the serum from a person, which has everything else in it, you just take antibodies just against the virus and use that. And then also, this is actually supposed to say interferon beta 1 beta, which is something that the virus now is known to target. So that's part of our immune response to viruses. And this virus actually targets that. And so there's some promising results showing that if you actually use it, it has to be at the right time, that it can actually have some positive effects. So in terms of limiting the damaging inflammation, you've probably heard about dexamethasone. It's been all over the news. So that's looking promising for the late stages. And there's also, this is a drug that's used for rheumatoid arthritis, IL-1/6 inhibitor, and that targets the cytokine storm that Dr. Harvey talked about this morning. So it's to minimise that. So again, a lot of really cool stuff going on here. So, you know, we're going to hear, I think, you know, over the next few weeks and into the next few months, lots of news coming out about all of these and many other things. Okay, we will move to the next slide. All right, so big question. You know, you might think this research is slow. Well, actually, it's really fast compared to most research. That's because so many people are working on everything. But, you know, the reason that it's slower than most people would like it to be is that there's many steps to doing research. You have to first do the basic research to figure out the biology. Then you have to take it in, you know, show it works in cell culture. Then you have to go into an animal model and then humans. You know, obviously, if you just go straight to humans and it doesn't work and it's toxic, then you're going to create all this harm. So you have to go through those steps. Another problem is that because this, because there is so much panic and desperation, and so many people are jumping into this research area, the reagents are running out, and you know, people are doing really small studies, they're doing them too quickly, they're not controlling them properly. You've got non-experts jumping into the field like me. We are collaborating with a virologist, so even though I'm not an expert, you know, I'm consulting and working together with someone who is, but not everyone's doing that. There's also been very poor interpretation by the media of some of the things that have come out, and that's kind of been confusing and mixing everything up. There's also, you know, an anti-science movement in some circles, and so, everyone's been very careful that before, something is actually used in patients, that it's actually going to be not just effective, but also safe. And then there's the production and distribution issues. Okay, I'm going to talk really fast about viral survival because I want to get into the eye end of this. So virus survival depends what the surface is, how long the virus survives on it. And in general, you can think about it this way, that the more porous the surface is, the shorter the survival time, and the harder and smoother the surface is, the longer the time is. And there's many theories as to why that is, but no one knows for sure. Dr. Harvey mentioned just soap and water is fine for washing hands. For inanimate objects, you can also use heat. Heat, works well. UV can work. Peroxide is also very good. So there's many different choices for inanimate objects. As Dr. Harvey mentioned, this is a very fragile virus. The envelope is not very stable. It doesn't survive very well outside of cells. Okay. I put down a whole bunch of other little tidbits that I thought were really interesting, some of which you may already know and others not. Okay, so we know transmission's low outdoors. Why is that? Well, it's not just the that we've got less confined space. There's also some evidence that vitamin D levels are important, which could also explain why that, with some viruses, there's a seasonal effect. We tend to have higher vitamin D levels. And then there's also a study that came out showing that sunshine kills the virus directly. So if you get out in the sun and, hang out there, somebody spits out this virus, the sunshine's going to kill it. So that's good news. So good to stay outside. There's a hormonal influence. This is coming out. There's some thoughts that progesterone levels may be important, estrogen and also testosterone. So there's evidence for all three of those things as being important, which could explain the age factor. It could explain various other things about, men being more susceptible in certain age groups. Children are not just less susceptible to the disease, but it's also becoming clear they don't transmit it very well either. So children do not give it to other people as well as adults do. So they'll get it from someone, but they don't give it that easily. And there is some emerging scientific evidence that the receptor for the virus may be less expressed in kids. There are not just super spreaders, there are some extreme super spreaders. So this is more so than other viral infections and nobody knows why yet. N95 masks. Okay, it's the reason to not wear these is not just because they're needed in some circumstances and you don't want to hoard them, but actually they're not great for preventing transmission. So they might protect you when you wear it, but they're not very good at protecting other people. And that's because if you sneeze wearing an N95 mask, the sneeze, everything just comes out the side of it. If it's fitting, it should be fitting properly. Okay, as I mentioned, the virus is vulnerable and it's not trivial to grow. And so a lot of the studies that have come out showing that the RNA is everywhere doesn't necessarily mean that there's viable virus everywhere. Okay, so people have been very misled by this. Like for example, in the cruise ship, they showed where everyone was getting infected, they showed that the virus survived for 17 days after no one was on the ship anymore. Well, it turns out that that's because if the RNA will just hang around for a really long time, but that doesn't mean that there's actually viable infectious virus. The infectious dose of this thing is completely unknown. We have no idea. Although we do know that for SARS-CoV-1, which is with the original SARS, it was about 300 particles. There's turning out to be some genetic susceptibilities. So blood type A, which I'm A+, which I used to be proud of, that I had an A+. It was the only A+ I've ever had. But apparently that does make you more susceptible. And there also seems to be something on chromosome 3 that seems to enable susceptibility. But nobody understands the details of that yet. The virus has been found all over the place, not just in the lung, can be in the gut, in the brain. They've found it in the optic nerve in some patients and elsewhere. You've probably already heard about this. I'm not going to go over that. This is something you may not know. Coins are antiviral. They basically explode the virus. So if you go to the ATM and you don't want to touch the buttons, you can use a coin. And then after that, the coin will not be contaminated. Also something else, we don't hear much about this because we don't want to know this, but it turns out that the US has been a major contributor to outbreaks in other countries. In fact, in Israel, they've shown that about 70% of cases can be traced back to somebody who brought it over from the US. All right, so now I'm going to talk about the eye for about the next 20 minutes. Okay, we don't know a lot about this, so some of what I'm going to talk about, actually a lot of what I'm going to talk about is based on what we know about other things, and some of it's going to be based on what we know about anatomy, and other things are just going to be pure speculation. I'm also going to tell you about some of the research results that we have, and there are a couple of published studies now. Right, and what I'm going to do is I'm going to address 4 questions. And these, I think, are the big questions that everyone's asking about the eye. So the first one is, can this virus infect the eye? The second one is, and this is a completely separate question, can it use the eye to access the airways? So what I'm talking about here, I'm not talking about, well, if it's infecting the eye, can it then go to the airways? I'm talking about, can it just go through the eye to the airways without infecting the eye at all? Okay, the third question is, okay, someone's got it on their eye, whether it's actually causing infection or it's just contaminating their eye. Can it then spread to someone else? Okay, third question. This is the fourth question. So given what we know, what we don't know, what we think and what we're speculating, what are the issues around spectacle and contact lens wear? And I'm going to tell you some things that may differ from what you've heard elsewhere. All right, so in order to think about whether this virus can actually infect the eye, that was question 1. The first thing we need to think about is, can this virus actually infect ocular surface cells? All right, so I'm just going to torture you with some basic science, but very, very briefly. All right, so there's the virus. When it infects a cell, it binds to ACE2. In order for it to enter the cell, the cell has to also express this. This is a protease. This protease modifies the interaction between the virus and ACE2 and allowing the virus to be taken up into the cell. Okay, if the cell doesn't express this, it doesn't matter that the cell expresses ACE2, it's not going to go in. Okay, having said that, there's a second way that this virus can get into cells, and that is through endocytosis. So cells just randomly eat stuff, right? They just take things up into endosomes. Okay, so the virus can get into the cell in that way. But in order for it to get out of there and not be killed in the cell, it has to express this. Okay, so this is cathepsin L. If cathepsin L is expressed by the cell, the virus can use that to actually get out of the endosome and end up in the cytoplasm of the cell. So that's a second entry pathway that the virus can use, even if ACE2 is not being expressed. Okay, so now let's talk about what we know. The ocular surface cells are equipped for virus entry. That's been shown. It's been shown from studies, this is not viral research. This is just research looking at ocular surface cells where people have looked at what they express. And the research shows that ocular surface cells express all 3 of these things. And in fact, the most strongest expression is actually in the superficial conjunctival cells. But what we don't know is whether the virus can actually get into the cells. So showing that the cells have these 3 things means that potentially the cells could take up the virus, but we don't know this for sure because no one's ever done the experiment. Okay, we're gearing up to do this experiment. We're collaborating with another lab here at UC Berkeley, Eva Harris's lab. In order to do this research, you need access to a BSL-3 facility. There is one on campus. Eva Harris's lab uses that for their virology research. One of the reasons we don't have the answer to this question yet is there's hardly any BSL-3 facilities around the world, and they're currently being flat out used by all of these researchers that are trying to work on this virus. So this is not a trivial experiment to do, even though it sounds like it should be. But we're going to be doing that. Okay. All right, so now I've just talked about cell cultures. All right. What do we know in people? Can this virus infect ocular surface cells? Well, we do know that conjunctivitis occurs in a small percentage of COVID-19 cases. Okay, so it's been estimated that it's, maybe 1%, although there was a study came out saying 30%. But the thought there is that most of those people just had, red eyes because they were sick, as opposed to actually having conjunctivitis. And it's probably closer to 1% of cases. So it's not a major site, but what we don't know is whether the virus is actually in there just because they have that conjunctivitis in most of these people. However, the virus has been isolated from the ocular surface of some patients with COVID-19. And when it was isolated, that virus that was taken from the eye was then shown to infect cells in vitro, not ocular surface cells, but cells that they're able to take up this virus. So that shows a direct connection. So you can get the disease, and someone with the disease, you can isolate the virus, and then that virus, you isolate it and it can go and infect cells. So that basically, to me, suggests that it can actually infect the ocular surface. What about asymptomatic people? Well, we don't know, because they've not been tested. So nobody's actually taken asymptomatic people and just sampled their eyes to see whether the virus is in there. Okay, so quite big clinically relevant question number 2. This is clinically relevant, not necessarily for us as optometrists, but in terms of patient safety and public safety. That is, can the eye be a mechanism by which the virus can get into the lung? Even if it doesn't infect the eye, can it just go through the eye into the lung? All right, so a different question. To think about this, we have to consider the relevant ocular anatomy. So now those of you who were in one of my lectures over the last probably 20 something years, in first year will have recognised this slide, deja vu. This is the tear drainage system. So I'm not going to go through the details of this, but you know that the nasolacrimal drainage system goes through to the back of the nose and into the throat. And that the tears are, this is happening all day long. You've got about 1 to 2 microliters being produced every minute and going through this system. And not only that, but this is actually an active process. So blinking drives this drain. You remember this? So you blink, when you blink, close your eyes, it constricts all of the whole nasolacrimal duct system. And then when it opens up, it sucks the tears in. So there's actually an active pumping process for the tears to go down. So this is actually sounds even worse than the nose, and in terms of possibly transporting the virus. Because if you get the virus in your nose, there isn't actually an active transport system for the virus to go down, like there is in the eye. So theoretically, if you have a particle that goes on into your eye and it's small enough to get into the tear drainage system, it's going to end up in the back of your throat. We all know this when we put eye drops in the eye, and you can taste it shortly after as it goes down. Okay, so now I'm going to tell you about a study you probably don't know about that provides what I think some evidence that this actually the eye could be a portal of entry. Again, this paper is not published. It's currently under review for Nature, a really top journal, Nature Communications. If it gets published there, I think it's going to be all over the news. I've looked at it and it looks pretty good to me. So what they did, they actually took monkeys, so primate, because you can't do this to a human. They put the virus onto the ocular surface. This is the only downside of this study. It's very small. They only had a couple of monkeys in each group. But of course, when you work with monkeys, you're not allowed to use large groups of animals. So that was necessary. What they found was that the virus went to multiple locations, including the lower lung. In the lung, there was classic lung pathology, the same type of pathology that was seen when the virus was introduced intratracheally, and very similar to what you would expect in a person. There was also a serum antibody response that happened, and actually looked even stronger than when it was introduced into the trachea. But importantly, that virus actually disappeared from the ocular surface after 24 hours. So it was no longer in the eye, but it was in the lung. And this continued for several days, and the virus was continued to be isolated for several days after that. Okay, so it sort of transitioned through. I'm going to skim through this really fast, just some of the data. This basically just showed that the footprint of the virus, its RNA, actually aligned with the tear drainage pathway. So all of the tissues that the RNA was found in aligns with where the tear fluid drains. The chest radiographs that actually showed the lung disease, in the top is the conjunctiva versus in the bottom the trachea. And this is the histopathology, again, also showing inflammation. It actually showed the viral antigen was there, not just the RNA, all along the tear drainage pathway. And here's the serum antibody response confirming the disease happened. The red bar there, that's actually the conjunctival inoculated. Have a look how large that bar is. The trachea was the dark green. So there seemed to be an even larger antibody in the serum antibody response after it was introduced on the eye. And this is just the data showing that the RNA actually transitions away from the ocular surface on the left and ends up in the airways. Okay, so that's a monkey. What about humans? Well, again, you can't do that study in humans. Okay, so what we've been doing is we've been thinking, well, what can we actually do in a human to explore this? So we thought, well, okay, so if the virus is going to go down the nasolacrimal drainage system, it would have to survive in tear fluid. So we thought, okay, this is an experiment we can do. We actually had some tear fluid in the freezer, human tear fluid that we were using for our bacterial infection research. And we've been able to get that tear fluid and use it to challenge the virus. Because we thought, well, if the human tear fluid can kill the virus, then that would be good to know, because there could be therapeutic potential. But if it doesn't, then that would again lend more evidence to the, or not evidence, no, it wouldn't prove it, but it would lend some credence to the possibility that in humans, the virus could use the eye as a portal. So we did this experiment in collaboration with Eva Harris's lab. What we did was we took the virus, added it to human tear fluid for 30 minutes, which is how long other things have been, that's generally the time people use when they're testing things for activity against this virus. And this was then the plaque assay, the conventional plaque assay that's used to test for viral survival that everyone's using, studying this virus, to count the surviving virus. And here's the results. And I'm going to show you this, and no one's ever seen this before, so you're the first people to see it. We haven't posted it anywhere, it's not been published, and I've not presented it. This was done last week. We compared it to peroxide. So what it showed was that there was unfortunately, the results came out equivocal, meaning that it doesn't completely kill the virus, but we can't say it has no effect. There was a small effect. This is a log scale. The efficacy of tears was, roughly about the same as, say, 0.003% peroxide, which was so we had different concentrations of peroxide as a control. In a second experiment, what we did was we added the virus to tears, and then we looked at the ability of the virus to infect cells, as opposed to just looking at its viability. And that, again, showed a very, very small effect. That was actually statistically significant. It's a log scale. So it was about half, again, showing about the same as 0.003% peroxide, which is not very much. So very little activity, nothing to get too excited about. We're doing some other experiments now to see if tear fluid can actually change the capacity of cells to interact with the virus, because we haven't done that yet. Okay, so taken together, for this question of whether the eye can be a portal. Okay, so here again, I'm just going to summarise. The virus has been isolated from the human ocular surface. When it's isolated, it can infect cells in vitro. Okay, tear fluid drains into the back of the throat. We know that from the anatomy, and the virus can survive in human tear fluid. Okay, the virus can be used as the virus can use the eye as a portal of entry in monkeys. That's been shown. Even if it's no longer on the ocular surface. So you culture that ocular surface, you don't see it, but it's still used the eye as a portal. So what that means, and the reason I'm belaboring this point is because there's people out there who are saying that because the virus has not been isolated from the eye, it necessarily means that the eye is not a portal of entry. But I would argue that not showing the virus on the eye doesn't tell us that it's not a portal of entry. And actually, from everything that I know and have seen, I'd be very surprised if it wasn't a portal of entry. It doesn't mean it's the main portal of entry, but I think that we need to be considering that as a strong possibility. All right, so now the third question, the clinically relevant. Can this virus infect someone else if someone has it on their eye? This is an example of not socially distancing, eye-to-eye contact. I'm guessing that it could. Firstly, because ocular surface isolated virus can infect cells in vitro, so why wouldn't it infect in vivo? And also there's some studies have come out and showing in New York that the ophthalmology residents in New York were having a very high infection rate compared to other specialties. And in fact, it was in the same category as people in emergency medicine and anesthesiology. And so both of those things to me suggest that. Okay, so what can we do to minimise these three things? The actual infection of the eye, eye being used as a portal, or the eye transmitting the virus. Okay, so first of all, we have to think about how the virus would get into the eye in the first place. So, you know, one way obviously would be touching the eye with using a finger or an object. As Dr. Harvey mentioned, surface transmission is not thought to be so much of a major player. Droplets, I think, are going to be the main one, from sneeze, coughing, talking, sneezing. These follow a straight path, importantly. Okay, someone coughs it out, sneezes out, straight path, falls to the ground, usually less than 6 feet. So if you're beyond that, further away, it's highly unlikely that you're going to get a droplet on your eye. Aerosols, okay, these are the small, really tiny particles. These can float around. You know, people were concerned, well, maybe they'll get around my glasses or whatever. But what the research has shown for aerosols is actually don't land on slow-moving surfaces, and that's true even if that surface is wet. So I confirm this. I actually talked to an expert who's an aerosol physics person who, and I had a long conversation about this. Could it land on the eye? And this person seemed to think no, that aerosols are not going to be able to land on the eye. Probably not on your glasses either, or your contact lenses. So really it's the droplets that we really need to be thinking about. Okay, so what if you wear glasses? All right, so if you wear glasses, I think that while you're wearing glasses, you're probably going to touch your eyes less. You might touch your glasses, but you know, probably better to touch your glasses than your eye. Aerosols, we don't need to worry about that, as I just mentioned. Shouldn't land either on the eye or the spectacles. The droplets, okay, if you're wearing a pair of glasses, that droplet's going to probably land on your glasses and not on your eye for almost all angles of projection. You would have to have your head in a very specific angle with that droplet coming in from the side in order for it to land on your eyeball. So if you're wearing glasses, you're probably going to be protected against droplets. The caveats of wearing glasses, okay, I'm telling you here, you know, it's kind of obvious from what I've just said, that I'm implying that wearing a pair of spectacles could protect you. The caveat is not going to be as good as PPE, okay? So there are some people who have been saying that, you know, glasses aren't protective because they're not PPE. Well, just because they're not as protective as PPE doesn't mean they're not going to protect you at all. But again, consider they are not PPE. If the spectacles get contaminated, the virus survives well on hard surfaces. So some people have brought that up as an issue. But there's a very simple solution to this, and that is you just wash your glasses with soap and water when you've come back from wherever it is that you were also wearing a mask. You take off your mask, take off your specs. You have to wash your hands anyway, so you might as well wash the glasses at the same time. Okay, what about contact lenses? Do they impact risk? Well, there's no data for any virus, let alone this virus, for either eye or lung infection, that contact lenses impact risk. Potential positives of wearing contact lenses, well, maybe people who wear them might wash their hands more often. It can be easier to wear a mask than with glasses. Wearing glasses might put someone off wearing their mask. But potential cons, I'm kind of just speculating, I'm trying to cover every base. You know, wearers might touch their eyes more often if they're wearing lenses. And it might discourage protective eyewear if both the contact lenses and specs have the prescription. And there's many other questions that we don't know the answer to, which I will not go into because we're running out of time. And I want to make sure I don't go over too much. But you're going to have a handout and you'll see those points on there. So is eye protection a good idea? In the clinic, absolutely. There's a study that's come out showing that the eye protection actually does reduce infection rate. It was a meta-analysis of many studies. This was face shields or goggles. For the public, it's probably unnecessary if you're socially distancing because you're further than 6 feet away, you're out of range of droplets. But if you're not socially distancing, so this is the same time that you'd be wearing a mask, it's unlikely to hurt and it might help, even if it's not as good as PPE. So some people already wear glasses. What about those who don't? So this would be contact lens wearers and emmetropes. People could wear either sunglasses or plain specs, or somebody could develop an eye mask. There's an opportunity here for industry, for something that's disposable, could be reusable, easy to clean, could be a fashion item, and it could be used over specs. So this is something I've been really pushing for, that I think we need that. I'm going to probably finish on this slide and not go into the next couple, but you're going to have these on the handout. On the handout, I encourage you to read the next slide because I want to finish on a positive note. Unfortunately, a lot of people are dying from this pandemic, but I think a lot of good things are also going to come from this. We're seeing an acceleration of discoveries, like the world's going crazy with biology and science. You know, people are becoming more educated about science. You know, they're watching CNN, they're learning about infectious diseases and about the value of research and of healthcare. And we're going to see application to solving many other problems. There's a lot of removal of administrative barriers. Scientists are learning about cooperation, so there's less competition, more appreciation of collaborations. And we're also understanding the importance of preparedness so that when something else happens, we're going to be in a better position to do something about it. So I encourage you to read this on the handout. Very positive comments from researchers in the field. And also, I'm just putting up a slide here, which again, you'll have on the handout. These are additional resources. I encourage you to read widely on this and keep up with it. What I've included here is not just the Academy of Optometry's website, but also the Academy of Ophthalmology's website. And I encourage you to look at that because there are scientists who work in virology and who study ocular surface virology who have written some very good articles there. So please don't just limit yourself to the optometry website. There's also, this is a fantastic program, the UCSF Grand Rounds program, if you want to learn more about the medical end of it. And then the American Society for Microbiology has many resources. These podcasts are meant for non-scientists, This Week in Virology, and you'll learn lots of really cool stuff if you listen to those podcasts. So at this point, I apologise for running over, but I'll be happy to answer some questions. Okay, so the first question, I'm going to read it. Some studies show that the average incubation period is 5.8 days and the serial interval is 5.2 days. Okay, viral RNA is detected about 20 to 21 days after onset of symptoms. What is the scientific basis for quarantine of 14 days for those who test positive or those who have been exposed? Why is the quarantine duration not longer? Okay, so this comes down, I was just listening to one of those podcasts I was telling you about yesterday, and someone was talking about that. So this comes down to the RNA, detection of RNA does not necessarily mean that there's viable virus there, okay? And that's because the RNA is just little pieces of the genetic material inside the virus, okay? That's not necessarily telling you that there's actually an actively viable viral particle there that could spread to someone else. Okay, should we be worried about, actually I would encourage you to listen, it was the June 17th podcast from This Week in Virology on the ASM website. So if you want to, because they actually discussed this in a lot of detail about antibodies and also antibodies and also the RNA results. Should we be worried about aerosol spray with NCT? Aerosol, okay, so aerosols, okay, so NTC. Okay, well, remember that, so aerosols are an issue only in terms of the lung disease, okay, because you can, I mentioned that aerosols are not a problem for the eye, but you can breathe in aerosols. So they don't land on surfaces, but when you breathe in, or you breathe in through your mouth, you can actually suck in aerosols. So aerosols are an issue, not for eye as the portal, but they're an issue for the nose and mouth. So if you're spending an extended amount of time or being close to someone and there's aerosols there, then that could be an issue. Will wiping tonometer tip with alcohol be sufficient for disinfecting? Yes. So 70% alcohol is sufficient. Again, it's a very fragile virus, and so that will work really well. Okay, second question. If there are any other questions? Okay, so if there's no other questions, again, I'd encourage you to really keep abreast of this and to really think about these issues, because every day there's more and more coming out. And I wouldn't just watch CNN or Fox News because a lot of what the media is talking about are not necessarily those things that are the most critical for us as optometrists and people working in healthcare. They're often things that attract news, attract viewers. And so, definitely please keep going there. And I'll also qualify that anything I said today could turn out to be wrong as we learn more and more, because every day there's more and more information coming out. So anyway, I thank you very much for your attention.
Thank you, Dr. Fleiszig. We will now take a short break and resume at 2:10 p.m. Pacific time for our last lecture of the day.